All right, everybody, let's do photosynthesis part 2. So here we're going to talk really about a lot of the things that happen inside photosynthesis and how there are certain factors that limit the amount of photosynthesis can happen. So let's outline what a couple of those things are first by talking about the overall equation for photosynthesis. So for photosynthesis, the plant needs some carbon dioxide, it needs some water, and it needs a little bit of light. And we talked about this. The light is used to break apart water molecules, and as a result, some O2 is produced. The light is used to break the water apart, and O2 is produced. This is a waste product. The plant doesn't really need it for the most part. We'll find out why it uses some of it later on, but for right now, we just need to think about it as a waste product, and it goes up into the atmosphere, and we use it. But the plant also makes a lot of carbon compounds. And that's the whole point behind photosynthesis, is to make carbon compounds. Now, we always say that the plant makes glucose, and that glucose can be used to make cellulose for the cell wall. It can be used to store as starch, so the plant can use that for energy later on, or it can just be used as energy in the cell. But plants don't just make glucose. Think about it. You eat foods with olive oil and vegetable oil and canola oil. Those come from plants, so the plants also use this process to build carbon compounds like oils, which are lipids. And also, plants make proteins, and proteins have carbon in them. You know this because certain plants, like legumes, peanuts and beans and things like that, contain proteins. All my vegetarians out there, you know this. And so some of the carbon compounds that are produced include proteins as well. So we mostly focus on glucose because we think about the relationship between energy and photosynthesis. Okay, so there are a few factors that limit how much photosynthesis can happen, and this is part 2 of your drawing quiz. You all have to draw the absorption spectrum for chlorophyll A, and make sure you understand that chlorophyll A is the main photosynthetic pigment in these plants. All right, so you can draw that absorption spectrum. You're also going to draw one of 3 graphs of factors that can affect the rate of photosynthesis. Those 3 things are temperature, carbon dioxide concentration, which is kind of like a substrate for photosynthesis, and light intensity. All right, so let's think about our graphs here. Now, since photosynthesis is a really enzyme-driven process, it's easy to think about these graphs as sort of similar to our enzyme graphs. So let's draw graph number 1 here. On the x-axis, we need to have temperature, and we're going to make that in degrees Celsius. And on our x-axis, we're going to have rate of photosynthesis. Now, there's all kinds of things we can do to measure rates of photosynthesis. What are some of those kinds of things we could do? We could measure the amount of oxygen produced. And there's a few ways we could do that. We could use some sort of digital meter that can detect oxygen, or we could maybe put a plant in some water and watch the oxygen bubbles bubble off of it, and we can count the bubbles. We can measure the amount of CO2 consumed. And this one is a little more difficult because you have to enclose the whole experiment so that no extra CO2 gets in there. That one's kind of tough. Or you could measure the amount of glucose produced, and that one's really hard. I mean, how do you get inside the plant and measure the amount of glucose that's been produced? I would say this one's probably the easiest one to do, and it's something that we're going to do in class as well. So for this graph, though, we're just going to think about this not as any of these 3 right here. We're just going to say, how much photosynthesis is it doing? All right, just like our previous graph for enzymes, this starts off slow. So we should have a nice steady increase, but there's going to come a point. There's going to come a point where all of those enzymes that are involved in photosynthesis can't deal with that temperature, structure of the proteins break down, and the enzymes denature. So therefore, no photosynthesis can happen. So I want you to think, and I want you to go back in what I just said in this video here. I want you to think about how you're going to annotate these 3 points on the graph based on what I just said. Okay, that's graph number 1. Graph number 2, carbon dioxide concentration. So let's do carbon dioxide concentration on the x-axis. There's all kinds of ways we could measure carbon dioxide concentration. Maybe we measure it in parts per million, which is a really common way to measure the amount of gases in an atmosphere. Once again, on the y-axis, we have rate of photosynthesis. So carbon dioxide is a requirement. It's a requirement for the process of photosynthesis to happen. And so if there's no carbon dioxide there, not a lot of photosynthesis happens. And so our graph starts off really slow. And as we increase the amount of carbon dioxide concentration, more and more photosynthesis starts to happen. But because this is an enzyme-driven process, there's going to come a point in time when you can't fill the active sites up any further. These are totally saturated. They can't be filled up. They're all full. And then as soon as they empty, another carbon dioxide goes right in there. So you can't do photosynthesis any faster. So once again, there are 3 points on this graph. Go back in the video and think about how you're going to annotate these 3 points on the graph. And finally, we have our last graph, which is light intensity. Light intensity. I don't know about you guys, but whenever I've gone shopping for light bulbs, which I feel like they're burning out all the time, so I'm buying them all the time, I always see light bulbs as being labeled as having a certain amount of lumens. So let's say that that's our unit here. Once again, on the y-axis, we have rate of photosynthesis. Now, light isn't really a thing you can pick up and hold, so there's not a concentration of it. There's just an intensity of it. But this is a process. I mean, this is a process that's driven by certain things, and when you don't have a lot of one of these things, it doesn't work very well. So the same as the previous graphs, when you don't have a lot of light, you don't have a lot of photosynthesis happening. Remember that you need the light to break down the water. You need the light to break down the water, which releases the hydrogen ions that photosynthesis uses later on, and oxygen is released as a result. So when you don't have a lot of light, you're not doing a lot of that breakdown of the water, and photosynthesis needs those hydrogen ions. Why? That's what you'll learn in HL. And then, as we get higher and higher and higher, we're going to get a point where you can't make photosynthesis happen any faster. And so the same as carbon dioxide concentration, the same as substrate concentration, this graph is going to level off. So what goes here? What goes here? And what goes here? Go back in the video, listen to the kinds of things I was saying, and think about very short statements that you can put down that describes what is happening in these points. You shouldn't be writing a single word down, like optimum rate. You should be adding a little something to that. It's the rate at which photosynthesis does not go any faster. That's not a good one at all. It's the point at which photosynthesis doesn't go any faster, or something like that. Okay, that's it for this one. What we'll be doing in class with photosynthesis after this is learning about some procedures to determine the rates of photosynthesis and starting the first 2 parts of our IA. Have a great night.